friends, welcome to another video tutorial from Shomu's Biology. And in this video lecture, we want to talk about eukaryotic transcription. And actually I've made a video about eukaryotic transcription almost 3.5 years back. So this time I'm making a video about the eukaryotic transcription from a different angle. And eukaryotic transcription is more complex compared to the prokaryotic transcription. And it's always advised that you should understand prokaryotic transcription first, then come and watch the video of eukaryotic transcription. If you want to know about prokaryotic transcription, you can watch those videos in my channel itself. The link of the videos are provided in the description, as well as the playlist link is also given. But eukaryotic transcription is complex because in eukaryotes, during the process of transcription, there are two components of the DNA that needs to be transcribed, introns and exons. And among both of it as well, the eukaryotic gene is under the regulation of one promoter. So one gene under the regulation of one promoter. We call it monocistronic gene expression. While in case of prokaryotes, multiple genes can be under the control of one promoter operator complex. This is known as polycistronic expression, or we also call it operon concept. So in eukaryotic transcription, the most difficult part is the start of transcription itself, that is the initiation of transcription. Because in prokaryotic transcription, we see the beginning of transcription is carried out with the help of a factor known as sigma factor. Now that sigma factor is nothing but a type of transcription factor. But in eukaryotes, we will see there are so many different types of transcription factors utilized. In case of prokaryotes, there is only sigma. But in eukaryotes, so many types. And most of these transcription factors play important role in the initiation of transcription itself. So for our understanding, we divide the transcription into three different parts: initiation of transcription, elongation of transcription, and termination of transcription. Now in this video I am going to talk about the initiation of eukaryotic transcription. And among these three separate parts, you need to understand initiation and termination very well, because the elongation part is same between prokaryotes and eukaryotes, because in elongation that's simply polymerization of ribonucleotides. So RNA polymerization event, the mechanism of which is exactly the same. So let's begin with the initiation of eukaryotic transcription. Now there are two parts to the initiation that I'll tell you. The first part is the mechanism of how the transcription begins in eukaryotes, while in the second part I'll tell you the regulation of this transcription initiation in eukaryotes. So the first part is the mechanism, and in the mechanism part, the most important thing you need to know is the transcription start site. This is the very first important thing that a gene must have. Okay. Apart from that, there should be start site recognition proteins. Now what are the start site and what is start site recognition protein? Start site is also known as the promoter region, and that is true for prokaryotic transcription as well as eukaryotic transcription. And that promoter region carries a unique sequence. Now as the promoter has a conserved unique sequence, polymerase, which is the major enzyme for the process of transcription, identifies that promoter and can initiate the transcription. But polymerase, which is the third most constituent, let me write RNA polymerase. But this RNA polymerase cannot initiate the transcription on its own, because RNA polymerase cannot directly bind to the start site and start the transcription. It requires the help of other accessory proteins, which are start site recognition proteins and polymerase recruitment proteins, which will initiate the process of transcription. Now the most difficult part about initiating the transcription is identification of the start site. So let me draw the DNA. Let me draw the sequence here. Let's say this is the DNA sequence, and let's say this is the region, we call it promoter or start site. But the problem is, let me draw, let me extend it as well so that we can draw all these things together. So this is the promoter, this is the start site. But RNA polymerase cannot recognize it directly on its own. Okay, this is first problem. So it requires the start site recognition proteins to recognize the start site, then recruiting the RNA polymerase so that polymerase can bind to the start site. Now the second problem is, let's say now the polymerase is attached to the start site. So even though the RNA polymerase is linked to the start site, it cannot initiate the elongation, right? It can never initiate the elongation on its own. Because for the elongation, that means to add nucleotides to make RNA, it also requires a positive signal. That needs to be provided by some other factors. And this is also a step where RNA polymerase can be inhibited for the transcription. So this is a point of regulation as well, right? And who provide the signal to either activating or suppressing the transcription? Different protein factors known as activators and repressors respectively. So now these are the regulatory proteins. The fourth important constituent, regulatory proteins. So these are the four major constituents of transcription initiation in eukaryotes. Now let's look at the process in details and how exactly it takes place. So in an overview manner, I told you, like first the start site needs to be identified by the start site recognition proteins. And how do they identify it? Because of unique sequence, and that sequence usually is those TATAAT. This is the kind of sequence that is present, right? Normally stretch of nucleotide sequences, which is consensus, that means conserved by structure as well as its function between different organisms. Now once it's recognized, then RNA polymerase must be recruited. So we need RNA polymerase recruiting proteins, okay, which are also a part of the transcription factors. So these start site recognition proteins, RNA polymerase recruiting proteins, all these things together, all these are known as transcription factor, denoted with TF. Okay, and there are different examples of such transcription factors. Now transcription factors are specific and they are associated with specific type of RNA polymerase. Now in eukaryotes, we need separate RNA polymerase for transcription of separate RNAs. Because you know there are three types of RNA, mRNA, tRNA, rRNA. So for mRNA transcription, which we are talking about largely, is carried out by RNA polymerase 2. So the transcription factor associated with RNA polymerase 2 will be TF2. Similarly, if a polymerase 1 is involving in the transcription, TF1 will be associated. And if polymerase 3 is in the transcription, TF3 will be associated. So now what we are seeing, that once TFs help the polymerase to be recruited, then they need a positive or negative signal. So that positive or negative signal to the polymerase will be provided by these regulatory proteins. And we can divide them into two different parts, activators and inhibitors. So activators and inhibitors, both of which will interact to the polymerase. Let's say this is activator. So it will sound a positive signal and transcription will go on. Now it may also recruit a repressor that will signal negatively and it will not allow the activation, so it will be, transcription should be turned off. This is a simple overview of how eukaryotic transcription takes place. Now let's talk about the mechanism of initiation in details. So to understand the mechanism of initiation, let me again erase this portion and try to explain it from the beginning. Because you know, I am not very fond of remembering all the name of the proteins and exactly what are their functions in place. But for understanding a mechanism, you need to have a very clear big picture. That's why I provide you with an overview. Now let me draw the DNA again. And let's say this is the one. This is the DNA. And let's say again this is the start site. Or we can also call it TATA or Tata box, which has the sequence of TATAT, which is conserved. Okay. So now at the very beginning, what will happen is that a first set of transcription factor will be in place, and I'll write down the sequence of all the proteins that are involving in this side. Okay, so I'll write it from here. So the first thing is TF2D. This is the first protein that will be involved. TF2D carrying a specific, if we draw the structure of TF2D, it has a specific DNA binding region. This is known as TBP, Tata box binding protein. Okay, and another component known as Tata box binding protein associated factor, or TAF. So this TF2D along with TBP, which is a part of TF2D, and another fragment called TAF, can easily bind to the Tata box 2D. Okay, binds to the Tata box. It recognizes the Tata box, and the structure of TF2D is placed in a way it will modify the DNA, and it will create a slight bend in the DNA. So let me draw the bend. It will cause the bend in the DNA. So now the DNA will be something like this. Okay, this is the again the Tata box. Creates the bend. So this is the very first step. Once DNA is bent, that is another signal, and the second sort of transcription factors will be in place, that is transcription factor 2A and transcription factor 2B. Now what are the role of transcription factor 2A and B? Transcription factor A and B will help to recruit RNA polymerase 2 in the promoter region. So actually transcription factor B is acting as recruiting, RNA polymerase recruiting proteins. Okay, so it will bring, so let's say let's draw, this is A, this is B, and it will help the RNA polymerase to be properly recruited. Say this is the RNA polymerase to be recruited to the promoter region. So the third step comes the RNA polymerase. But the question is, RNA polymerase cannot bind to the promoter on its own. It cannot recruit itself on its own. So recruiting proteins help in recruiting RNA polymerase, but RNA polymerase need a escort protein, that is TF2F, which is remain attached to the RNA polymerase and brings the RNA polymerase, guide the RNA polymerase to bind to the promoter. Okay, so RNA polymerase is now attached to the promoter. Now what happens? This state is known as a pre-initiation complex. You know what and why is pre-initiation complex? Because if you look at this structure, at this particular stage, polymerase is in place of start site, so it can initiate the transcription, but it will wait for the proper signal, because this is a point of transcriptional regulation. So if positive signal is there, then only the RNA polymerase will escape the promoter and transcription will begin. If negative signal is there by repressor, then the polymerase will remain attached to the promoter and it will not escape the promoter, so the transcription will be prevented. That's the easy process of regulation here. So let's assume that it receives a positive signal with the help of activator proteins. Okay, we'll talk about the activator event and repressor event in details later. So let's assume it gets a positive signal. So once receiving the positive signal, it needs to remove the promoter. But this promoter escape of RNA polymerase is the indication of transcription start. But that thing cannot be done without the help of other activator proteins. And what are those? Those are also some transcription factors. Now that's why we come to the fourth type of molecules. Again, fourth sequence, and in this case transcription factor E and H. Okay, transcription factor 2E and H. Both this transcription factor E actually recruits transcription factor H. Now the beauty of this transcription factor H is transcription factor H has enormous capability, like 2 or 3 versatile activity. One is that it can act as a kinase. It can also act as a helicase. So these are some versatile feature of transcription factor 2H. And why this? We are talking about helicase and kinase, because if you look at here, at this point, polymerase is linked, all the other transcription factors are linked, but the promoter is closed. So the DNA that is present in the promoter are also closed. They are not open. Right? That means the complementary strands are closed with each other. So at this point, we need to open the strands from each other, separate the strands from each other. And that can only be done with DNA helicase. So here TF2H functions as that helicase. Now remember one thing, before the polymerase should remove, this helicase has another function. Because if the RNA, if you study the structure of RNA polymerase in eukaryotes, you will see that RNA polymerase contain a C-terminal tail. Because you know, if like every protein has two terminal, amino terminal and carboxy C terminal. So the C terminal of this polymerase needs to be, it forms a tail, an extension of the structure. Now this extension needs to be phosphorylated, and the phosphorylation of this C tail, which is also known as CTD, C terminal domain of the RNA polymerase 2, the phosphorylation of CTD by H helps in promoter escape by the polymerase 2, and that will complete the initiation. So for that, helicase, that is TF2H, also need to have a kinase activity, and it utilizes ATP hydrolysis, and actually it attaches phosphate group to the C terminal tail, and that will cause the release of polymerase from the promoter, called the promoter escape. It will migrate from 5' to 3' of the newly synthesizing strand of mRNA. This is the overview of how the transcription initiation takes place in eukaryotes. Now imagine one thing, at this particular point, these are all the sequential events of different transcription factors involved. But there is a regulation as well, and the regulation is at the position of pre-initiation complex. And I told you that I am also going to talk about regulation in this video. So now let's talk about it, because while the polymerase is in place to the promoter, the regulation needs to be done, and the regulation is done by either activator or repressor. So both activator and repressor can bind with the polymerase. Along with that, it can also bind with some DNA element. So it has two separate binding sites, DNA binding site and polymerase binding site. And where exactly it binds to the DNA? Upstream of the start site. So let's look at here, and this is the upstream region of the start site, and this is where we can see that a place for repressor binding. So now if repressor molecule is attached with the help of DNA binding region here and with the help of polymerase binding region with the polymerase, then it will not allow the polymerase to move forward. Right? There are two ways the repressor can prevent the polymerase. It can alter the structure of the polymerase. Upon binding to the polymerase, it can alter the polymerase structure, which will not allow the polymerase to remove and go from the promoter or release from the promoter. On the other hand, it can prevent the polymerase escape, that is a melting of the promoter region. So this melting is required because you know, while the polymer is bound to the promoter, both the strands are joined together. So we need to separate the strands. So this melting can be prevented, because without melting, polymerase cannot initiate the process of transcription. And this, once the DNA is melted, we are calling it as a open complex, and while it's blocked, we call it closed complex. So transition from closed complex to open complex is also very important, which can be prevented by this repressor. Now on the other hand, if activator is in this place, activator binds upstream control and upstream regulator elements that are present in the DNA, known as enhancers. So if I draw this picture, if I extend this DNA further upstream, you will see small regions in the DNA known as enhancers. This enhancer is a DNA element, and there are activator proteins that can bind to the enhancer with the DNA binding site. So let me just put it an arrow. These are the activators. So this activator also have a DNA binding site and a polymerase binding site. So with the help of this polymerase binding site, this activator can link with polymerase that can allow the initiation of transcription. That's how easy it is. Okay? But the question is, you may ask one question, that I am telling you that the enhancer element of the DNA, which is also known as upstream control element or distal control elements, all these things, those are distal and placed many 200, 300, 400 nucleotides upstream of the promoter sequence. So how exactly the activator or repressor can be in direct contact with polymerase? And without in direct contact with polymerase, they cannot initiate the process of initiation. So the answer to it is yes, they are placed distantly, but they need to be interacting. And for the interaction of activator with polymerase or repressor with polymerase, sometimes the eukaryotic DNA needs to be folded and forming loop. So they start folding and forming loop. So once the loop structure is formed, then they can bring the structures close to each other. Let me show you how the loop structure is formed. Here. So let's say this is the DNA. This is, say, this is the region of the promoter. Okay. And it can form loop like this. And let's say this is the upstream control element, or what we can say activator. Okay. Activator element. This is the promoter. So how an activator molecule, let's say this is the one, will be in contact with the promoter. This is the way, because you know activator molecule needs to be in contact with the polymerase, and if I draw polymerase here, let's say this is the polymerase. So now we can see easily that the activator molecule has a polymerase binding region with which it can bind to the polymerase. So this is why looping is required. Now sometimes activator molecule cannot directly bind with the polymerase. So in those cases, activator molecule and polymerase are attached with each other with the help of a third molecule. So let me draw here. Let's say this is the activator molecule, and let's say this is the polymerase. This is the polymerase. But they cannot interact with themselves directly. So there is a third party in the middle known as mediators. So this is known as a mediator, or it's known as a transcription mediator. So this mediator molecule can hold the structure of both activator in one hand and polymerase in other hand. That will also bring both activator and polymerase close to each other to initiate the process of transcription. Now there are these two types of mechanism of transcriptional activation, and one type of mechanism is transcriptional repression. Because if repressor is present, repressor will not allow the promoter escape by the polymerase. This is one way of repressing. Another way is that it will not allow the transition from closed complex to the open complex formation. But in case of activation, there are two types of activation. One is known as the recruitment, another one is known as the allostery. Both of them are activation mechanism. The recruitment process says that the polymerase is guided and the polymerase is loaded into the promoter, because you know without the recruitment or without the recruitment proteins, although polymerase can bind to the promoter, but the binding is not stable. So always promoter binds and dissociates, attach and dissociates. So with the help of those proteins, polymerase can now properly bind and initiate the process of transcription. This is one thing. This is known as the recruitment activation. While the other type of activation, that is known as the allostery, is a situation where an activator protein will be in contact with polymerase. And while an activator protein in contact with the polymerase, in that case, there are two mechanisms that are possible. One is that binding of activator protein with the polymerase will alter the structure of polymerase in a way that it will easily release and remove from the promoter. So promoter escape is enhanced. On the second hand, on the second other process is it can also allow the transition from closed complex to open complex. So the activator does the job which is reverse of the repressor's job. That is how the process will continue. In both this way, presence of activators is very much important to increase the transcription rate above the basal level. Now if activator is not present, it does not necessarily mean that transcription may not happen. Transcription may happen, but without the presence of activator, the level of transcription will be basal level, very less. Okay, that's the difference between the activators and repressors, and that's how eukaryotic transcription is regulated. Okay, and transcription initiation is regulated, and initiation is the most important part for the regulation, because once the initiation is there, the transcription need to be completed, right? So it's a rate determining step. That's why it's very important. I hope you understood the process of eukaryotic transcription initiation. In the next video, I'll talk about the transcription termination in eukaryotes and the regulation of transcription termination. Okay, so stay tuned and watch the video.